You can see the slime diffusing into the water that we thought was starch, but the iodine test proved otherwise. I smeared some on a microscope. Let's take a look. This will be unstained, and then we'll look for a stain that will make this visible. I'm anticipating a lot of bacteria, which are super duper small. So we're gonna have to go to the oil level, 1000x. So I've put a little saline drop on our slime specimen here. I'm gonna stain it anyways, because we know that methylene blue likes phosphates. So if we got bacteria in there, they should light up. I've added my drop of methylene blue. It's gonna dilute, and we'll drag a cover slip across, and I'll use my super secret science absorbent paper to pull off any excess so it's not a total mess, and we'll see what we can see. So this is the smear, and what you can see is that the methylene blue is pervasive throughout. There's that white clump, or whatever that is. And when we look in the microscope, we want to look at the edges. So we see a little air bubble there at the tip of the needle, but then you see that blue streak. And when we zoom in, we're going to see even smaller cells, and they are most likely bacteria that have picked up the stain, and they're quite dense. They're quite small. We can see here, zoomed in, that same bubble, and all those little flecks. Same bubble. I'm not so convinced these are bacteria. I'd need to do further testing, like gram staining or something like that, which is a particular kind of stain that bacteria pick up. There's gram positive and gram negative, and it stains them very, very dark purple or a bright magenta, depending on the nature of their cell wall. They could be bacteria, highly likely. I've put a bit of oil in the slide so we can really get a close look at those, whatever that debris is, or bacteria, or both. Further testing would be required. So the blue is the methylene blue, and we know that methylene blue likes to connect to negatively charged things because itself is positively charged. Okay, so I've mounted my iPhone onto the microscope. We're just gonna let this simmer and sit, so I'm not moving around it. You can actually see the bacteria are wiggling around, and they're going in little different directions. So this is actually the potato rotting. If you ate this right now, if I eat this right now, I'm gonna have a wicked case of food poisoning. This is why we wash our hands, this is why we cook our food, and this is why we keep things refrigerated, because it slows all the biochemicals down. What you're looking for in this video is the random, seemingly random motion and the different direction of motion of each of these little cells. We are actually at 1,000 times magnification right now. And each one of those things that are moving is an individual bacterial cell. You can see them wiggling in all different directions, and they're just gobbling up that starch that was sitting in those amyloplasts. So those amyloplasts could in fact be intact. All those little clear beads, we can see some of them up by the 1 o'clock position, and there's one over by the 3 o'clock position there. They could still be intact, but the carbohydrates within them are not. I'm convinced of that because our beaker didn't turn purple. I will dispose of this, but now you can really see them wiggling and moving. And they don't have to move very far because they're literally sitting in sugar. I mean, imagine being in a room full of food. To these bacteria, it's Willy Wonka's chocolate factory. Everything's made of chocolate. Everything's made of sugar for these dudes. And they're quite content. And they'll swim towards that. They have receptors on the outside of their cells and their membrane that detect sugars and carbohydrates, and they will literally move towards the carbohydrates. The process called chemotaxis. The longer we wait, the more we watch, we can see the bacteria moving. And there they are. Pick one to focus on. Keep your eyes on it. Where's it moving? Where's it going to move? I don't know. Take your time and you can see that there's different morphologies. Some of them are darker than others. Some of them are rod shape. Most of them are rod shape as far as I see, but there's little dots that are sphere shaped. Staphylococcus aureus is an example of that, like a staph infection. Have you ever heard of that before? Now we can see the bacteria moving. And the methylene blue that stained up those potato membranes really well the other day now help us to visualize these individual cells. And they're moving all in different directions. Each one of them is its own individual, most likely a clone from which it came unless mutations occur.